good morning, good afternoon, and good evening, depending on where you are joining us from in the world. I am joining from Toronto in Canada, and I know that my other colleagues are joining from different parts of the world as well. It's a pleasure for me to host this webinar today. I want to start by introducing myself. My name is Mosamo Shabela. I'm a professor of public health. I'm a trained medical doctor and a family physician at the University of KwaZulu-Natal. I am currently the Deputy Vice Chancellor for Research and Innovation in the university, and work very closely with colleagues that are in biotech, innovation, and essentially trying to support the building of the innovation value chains in our context, and thinking about how to make efforts around innovation to be relevant to our context and to be relevant locally, as you will see with the discussion today. This is the sixth in the series of the webinars on diversity and clinical trials. Some of you may have been able to attend the previous ones in 2022, but this is the first one in 2023. So I want to welcome all of you to the webinar. I want to welcome the panelists as well who have joined us. Kemi Williams, Derek Akpalu, as well as Patrick Akagbo. I will introduce you to them shortly, but let me take this moment also to just thank the organizers, Amgen, as well as Bauer Africa colleagues. There's a number of people in the background who work very hard to ensure that we are successful in holding these discussions. So we are going to get started with our first talk, and I would like to invite Ms. Kemi Williams for you to put up your presentation. Kemi Williams is currently running a firm that basically consults on clinical research, previously Vice President at 54Gen. She's got an MBA, and she's going to talk to us about frameworks and infrastructures that are needed to sustain diversity in clinical trials. Without wasting time, I'm going to switch off my video, Kemi, and welcome you and invite you to do your talk. You've got 15 minutes. Anyone who's got questions, please include them in the Q&A. We will take note of them. At the end of the presentations, we will have a discussion. Thank you very much. Over to you, Kemi. You're most welcome. Very excited to hear your presentation.
Thank you so much, Prof. Thank you so much, Prof. And thank you to Amgen and BioAfrica and the panelists and the other participants in this session for this opportunity. I'm excited to continue to contribute to this important conversation around enabling diversity in clinical trials in Africa. Over the last few sessions, as mentioned by Prof, we've talked about what clinical trials diversity mean in Africa. We've looked at it in the context of genomic and precision medicine. Today, what I would really want to focus on is how do we start to establish frameworks and solutions for promoting sustained clinical trials diversity in Africa, with the goal of seeing how we're making progress on this topic. So once we've agreed that this is important, we want to start looking at how are we making progress when it comes to trial diversity in Africa. But before I proceed, I want to show the Amgen disclaimer here. The views that I'm expressing today are all mine and not necessarily those of Amgen. And let's get started. So what I want to touch on quickly is really an alignment around what do we mean by trials diversity in general. And I'm sure many people on this forum, you already have this idea in your mind, but just to make sure that we are aligned and grounded in our understanding, let's explain trials diversity to be the deliberate or intentional inclusion of participants that represent the real-world patients in trials, and in so doing, accounting for all the demographic factors that you would need to take into consideration to ensure diversity: race, ethnicity, gender, age, sexual orientation, geographic location, education level, and socio-economic status. I call this out because most times when we're thinking about clinical trials diversity, most people automatically think just race and ethnicity. But these other factors are actually very important if we want to ensure that we're not excluding certain patient population from the innovative trials that we conduct. And we know that clinical trials diversity is a moral and scientific imperative for so many reasons. I'll touch on just a few of the benefits that we may all already know. It ensures that we can generalize the research results that we obtained. It advances science. It promotes health equity, and it reduces healthcare disparities. And so it doesn't matter whether you're in the US or you're in Canada or you're in Africa, anywhere in the world, once we allow for clinical trials diversity goals to be achieved, we can reap all of this benefit. But let's look at the African context here. I believe because of the current maturity of the R&D industries in Africa, and when I mean R&D, I'm talking about biopharma, biotech industries, and them being in their infancy stage, that Africa actually also has some unique benefits that it can accrue from participating in the Clinical Trials Diversity Initiative. I've highlighted some papers that I have started to talk about and describe actions that they are taken to enable more clinical trials diversity in Africa. But let's talk about some of these things that they've highlighted. Inclusion of Africans in innovative clinical trials that are done usually by sponsors from other parts of the world has been linked with increased capacity within Africa to provide evidence-based care and better patient outcomes on the continent. And so what it means is that when Africans are involved in these trials, they now have to learn more about the products. They have to understand, they get the right education and training on the process for conducting these trials, more understanding and training around the impact of the treatment or the diagnostics tool on the patient that they are treating. And what automatically happens with these investigators is that they start to get more evidence-based processes and clinical care approaches for managing their patients. And then, of course, we can then generate results in the trials that will support our clinical care practices. Beyond that, having clinical trials diversity that involve Africans would definitely raise research standards within the continent, lead to healthcare sector improvement, and provide the badly needed investment that we need in the continent. What I'm hoping is that someday, in some near future, that our R&D aspect within the continent will be well developed enough that we can actually start to conceive ideas around drug targets and pull them through the production and development pipeline all the way to regulatory approval and commercialization. But until then, our ability to engage effectively with other external stakeholders would allow for these benefits to accrue in Africa. But we do have unique opportunities that we offer to those who come into Africa to run their innovative trials. Africa has one of the most genetically diverse population base in the world, with the most variant sites per genome and the rarest genetic variants. And when we understand that for most diseases, right, the incidence, the severity, and in fact, the prognosis of these diseases are usually influenced by our genomic makeup, then being in a region where you have genetically diverse populations definitely would inform and advance sciences. Africa still has a large population of treatment naive patients. And so coming into this region to enable trial diversity by including more Africans means then we can speed up recruitment and hopefully potentially reduce clinical trials cost. So now that we've talked about, the importance of trials diversity, because it's always good to align on, why we're doing what we're doing, I would like to introduce some frameworks and solutions that we need to start to establish. We need to start to push forward for these conversations around clinical trials diversity to be a reality, a successful reality, I would say, in Africa. And I've laid out 4 sectors that we should look into: partnerships and collaborations, research capacity building, regulatory and policy frameworks, and patient-centric strategies. And what a thread that you will see as I go through this is that there is a need, and I think an important push on my hand as well, to start looking at how Africa is setting itself up as well to attract the type of partnerships and the type of innovations that we want, which we believe would lead to more clinical trials diversity objectives being met on the continent. So let's start with the partnership and collaboration part. We would now require that African stakeholders work closely with global or international stakeholders in terms of understanding the solutions that need to be put in place that meet our own local context. We need to look at partnerships between the private and public sectors as a way to also drive this initiative. How are we working with the academic research institutes? How are they working with private companies and private institutions in terms of helping to identify the unique needs within Africa when it comes to clinical trials diversity, and then helping to develop and implement solutions around those? Sponsors and communities, and for these in this case, sponsors are usually international or global sponsors who are coming to run trials in Africa, need to partner as well with the communities in which they are engaging. And so how are we leveraging relationships with community leaders, trade associations leaders? How are we engaging religious leaders? How are we educating patients to be sure that they understand what the clinical trials objectives, risk, and requirements are? This type of partnership would mean that we are creating the right foundation to be able to enable more trials diversity within the region. And what is important when we're thinking about partnership and collaboration as a tool for driving clinical trial diversity is to look at ensuring that the priorities and the values of the partners and all the stakeholders are well articulated and aligned. We want to ensure that we are working together, but moving in the same direction, not in different directions. It's one thing to have the right partnership and collaborations in place. It's another thing to have the right capacity to be able to do these trials. Government investment in the healthcare and research sector in Africa will help drive this aspect of our goals. We need the African government to start to understand that this type of funding really is another way to grow the economy within their countries. It's another way, in fact, to grow the GDP. And while they're doing that, which is helping the health of the nations, we are then able to engage more actively in all the clinical trials that are being brought into the region. And in fact, also start to design and implement our own trials effectively. Healthcare providers and research staff need to be educated and trained about what it means to find the right patients that represent the populations that are affected by the diseases, in fact, that they are doing trials on, but also to have the right tools for managing informed consent, for managing patient engagement. So an investment in educating our healthcare providers and research staff means that they become the liaison between the sponsors and the patients in helping to find the right people for the trials. We want to start looking at how we develop proper research facilities and infrastructures, labs, digital technologies that can be utilized effectively in trials, data management systems that would ensure data integrity. All of these requirements would help drive more and better recruitment of the right patients and trial diversity. And as we know that definitely in the movement in terms of precision medicine and trials diversity, being able to identify the right patients is important. and being able to monitor the outcome of the patient is important. So having an availability of advanced molecular diagnostics and expanding the availability of these molecular diagnostics become quite important as a tool for also driving trials diversity in the region. Let's look at regulatory framework. Some of the barriers to having trials being done in Africa has been related to the delay in getting regulatory approvals for clinical trials protocols. It will be important for our regulatory agencies across countries in Africa to start to develop transparent and efficient processes for clinical trials review and approval. It's also important that the agencies are working with policymakers to introduce guidelines and policies that would also promote clinical trial diversity objectives. And we do know that the agency staff across Africa can also benefit from training on good clinical practices and bioethics requirements, which would facilitate their review and support of clinical trials registrations. But let's not forget the patient as we look at these frameworks and solutions. We've talked about finding ways to engage patients more actively as another opportunity for enabling more trials diversity. So inclusive trial designs, where we're looking at inclusion and exclusion criteria that do not exclude certain people, maybe because of their biological variances, trial designs that incorporate cultural nuances and norms, so that we are not excluding people that may actually need important input from family members before they can be part of trials. What may happen in these cases when we have trials that are being brought in from other parts of the world, that there is opportunities to engage with patient advocates, advocacy groups, and also, in fact, investigators to modify the trial designs appropriately so that those trials can be effectively conducted and patients can be recruited in Africa. We should continue to educate patients on the benefits of clinical trials participation to deal with the mistrust of many years that have occurred due to the parachute research that the continent suffered from many years ago. And also sponsors need to start accounting for the financial impact that patients actually have to deal with when they have to be part of clinical trials. And so providing the right funding to minimize this adverse financial impact will be important. Some people have to leave their job or take time off unpaid to go to doctor's visit. Or some people have to figure out how to transport themselves for doctor's visit. These are things that we should build into clinical trials design as a way to soften the opportunity for patients to engage. And it's important that we start to look at sharing study results and providing drug access once the trials are completed in Africa. These are deep topics that we could take days to really dig deep into. What I've done is just provided a high-level overview of all the different things that we should be considering as we look at frameworks and solutions for promoting trial diversity in Africa. But I want to dig a bit deeper into the partnership and collaboration aspect as a vehicle for promoting clinical trial diversity. And the reason I want to do that is because I think that if we can get the partnership and collaboration aspect right, then they can influence all of the other frameworks that I've identified. And so when we're thinking about who the right stakeholders are that should come together to help drive clinical trials diversity, we should look at both local stakeholders in Africa and international stakeholders. So within Africa, we will want to look at healthcare providers, the research institutes, the regulatory agencies, the patient advocacy groups, the African government. And then we would look at international stakeholders like the biopharmas, the global health nonprofit organizations, regulatory agencies in developed countries, and professional medical associations and organizations. It's important that we start to look at creative partnering models, cross-country, cross-sector, cross-institutional partnerships within Africa, across Africa, and even externally with stakeholders outside Africa will be important. And as we come together and think about this partnership, it's also critical that we prioritize health equity and humanitarian goals. if we want to address the disparity that is associated with clinical trials, which also impact healthcare in general, and prioritizing humanitarian and benevolent goals become very important. And in that partnership model, we should treat local stakeholders as equal partners as well, so that the agenda of those partnerships are not driven only by external stakeholders' goals and missions. All in all, it's important to realize that when we start to reduce healthcare disparities in Africa by enabling more clinical trial diversity in the region, that in itself is a worthy return on investment. That in itself, it's a worthy goal to achieve. I won't spend too much time on this just because I am mindful of the time left. So I would just touch on a few actions, maybe one action per each stakeholder, that if we even start to engage in now, we can start to move the needle on clinical trials diversity in the region. For African government, I think they should start looking at introducing policies to attract more global biopharma presence within the region, rather than pushing these important partners away. What initiatives are we establishing so that they actually find Africa as an attractive destination for conducting their very important research? The Rwanda government is an example. They've been doing this already, establishing policies that attract biotechnology companies to set up base within the country. I would recommend that many other African governments start looking at this as a tool as well, not only for advancing healthcare, but as a tool that also supports clinical trial diversity goals. For African regulatory agencies, let's start to look at how do we streamline the clinical trials and drugs approval processes. There's no reason why trials that would take 3 months in other countries to get reviewed and approved would take up to 2 years sometimes within Africa. Sometimes those trials are already done recruiting in other parts of the world, and we're just beginning to recruit here in Africa. So looking at ways to partner with funding regulators to use shared learning and data to streamline our process will be very critical, once again, for attracting the right stakeholders to the region to do trials, but also to enable more development and research activities within the region. African research institutes play very key roles as well. They can help lead trial capacity building initiatives. Everything that I mentioned earlier around infrastructure that is needed, the African Research Institute can help drive those initiatives working across all of the stakeholders. But more importantly, by them demonstrating the benefit and the utility of clinical trials diversity, they can get government agencies to also buy in and provide the right resources towards those goals. Patient advocacy groups as stakeholders are important to the process, and we should not ignore them. They can help engage Ministry of Health and policymakers to enable that, to ensure that patients have access to trials, that patients are aware of the trials that are available within the regions. They can work with sponsors and investigators to design trials so that they are less burdensome for patients, and they're also relevant in terms of the cultural context. And they can definitely support patient education and recruitment. Care providers, by them getting more training and getting more educated, they are now enabled to be able to include a more diverse set of patients in their trials, cutting across different tribes, cutting across socioeconomic status, cutting across sexual orientation. And so enabling care providers, but also them stepping out and driving this mission will be quite important. And I just will touch briefly on our global and international stakeholders and the actions that they need to take to help drive this project, this initiative forward. Biopharma companies, it will be important that in the early stage, while they are thinking about developing a molecule or a target, that they start to prioritize Africa as a destination for early development activities for their clinical trials. Because once that is prioritized, that would go into the design of the trial, and it will go into how it is implemented, how those trials are implemented. And in fact, it would be helpful if they can also help fund clinical trials infrastructure development in Africa so that the region is more enabled to effectively conduct those innovative and sometimes complex trials. Global health organizations have been supporting already, though we need them to continue to support as liaison between all of these different stakeholders to help secure the funding and to help drive the capacity building activities. And in fact, we want them to also help encourage and support African-led trials so that we can actually have trials that are designed specifically for Africans, but that can also benefit the rest of the world. Foreign regulatory agencies can offer technical assistance and cross-trainings to their peers in Africa, and can also support collaborative inspections to help with product registrations. And we have professional medical associations, within the region, within the continent and outside the region as well. The ability for these medical associations or professional organizations to collaborate will be very useful to help with professional development and to provide opportunities to African investigators. And in fact, they can partner with local investigators to design and conduct context-relevant trials. All in all, we believe, and that is why I'm highlighting this, that ensuring more partnership and more collaborations across local and international stakeholders would really help us to start to develop sustainable clinical trial diversity solutions within Africa. So I would end this by saying that, why do we even care? Why are we talking about clinical trials diversity? Or why are we even talking about what it means for Africans or engaging Africans in the process? We know that where there is limited trials diversity, that there is that particular limitation is linked to high disparity in healthcare across populations. And that in itself is a form of inequality and injustice. And such inequality and injustice in healthcare is definitely very shocking and inhuman, just like Reverend Martin Luther King had quoted. And so to help drive diversity, we are also helping to close disparity gaps across Africa and across nations in the world. Thank you for this opportunity.
Thank you so much, Ms. Williams. You've touched on some really important points there. I think that the way you've captured the broad spectrum of stakeholders to engage and sectors to engage is quite compelling. In fact, one could assume that the primary stakeholder is biopharma, but we have demonstrated that actually there's a wide range of stakeholders to engage. I think there's a point you made that I would like to come back to later if you can start thinking about it. And it's this issue of the historical patterns of behavior around that have contributed to mistrust. And I suppose forms of exploitation, whether you think that there is an active intervention to be put in place to address that, and whether you think that there needs to be measures to prevent that from happening going forward. Your argument is very clear that we need to understand the diversity that exists in Africa so that as we develop interventions, especially around precision medicine and so forth, we have the infrastructure, the skills, we've got the genomics to be able to provide that level of care. Otherwise, we're going to fail to provide it. But the point you made around mistrust is something that I would like to come back to. Otherwise, thank you for that presentation. And I'm hoping that participants can also capture their questions for you in the Q&A, and you can feel free in the meantime to answer some of them. Let me turn to our second speaker, Dr. Derek Akpalu. If you can please mount your presentation as well. Derek is the CEO and co-founder of Revna Biosciences, and Derek has been previously with Johnson & Johnson for about 7 years in the US, but he's come back on the continent to set up this company. He'll be speaking to us around alternative practice models and partnerships to localize technology and advance molecular diagnostics. Over to you, Derek.
Thank you so much, Prof. And yes, thank you for the opportunity. And once again, a disclaimer. This session is obviously sponsored by Amgen, but please note that the views expressed here are not Amgen's, they are mine alone. So as the earlier speaker mentioned, the discussion on the Africa Bio platform with the sponsorship with Amgen about clinical trial diversity has been ongoing for a while. This is the sixth in the series, and at this point, the discussion is transitioning from the rhetoric or the focus on the issue to the how and the what. The how being, how are we looking to solve or address this topic that we all agree is of importance to us, and then importantly, what are we doing or how are we doing these? So my topic, my discussion today is going to be centered exactly on that. What are we doing at Revna Biosciences in the wider context of this discussion of increasing access to patient populations or clinical trial diversity in the context of the use of advanced molecular diagnostics locally to advance the agenda of trial diversity. So the topic of my talk today is going to be basically alternate practice models and partnerships to localize technology and advance molecular diagnostics using Revna Biosciences as the focal point, or to demonstrate what we're doing. And before I go into that, I think that one of the comments I want to make is that in listening to Ms. Williams' presentation, she did mention a few frameworks that I was happy to hear, meaning that from her perspective, our perspective, some of these are cross-validating each other. Throughout the presentation that I'm going to make, you will begin to see some parallels in what we're doing and what she eloquently put out as well in terms of partnerships, research capacity building, regulatory and policy updates, as well as the focus on the most important factor of all of this, which is the patient-centric focus or view. So on the revenue side, three main issues as it relates to clinical trial diversity and advancement of precision medicine on the continent concern us. The first is access to medicine and treatment failures. We know that on the continent, pretty much 95% plus of all medicines are imported with very little production locally. We also have a big issue with local diagnostic penetration. In some instances, about 80% of patients being prescribed medicines without an accompanying lab test. As you can see, in terms of capacity as well, even comparing sub-Saharan Africa to one country like the US, you can see how very inadequate the research capacity or diagnostic capacity is in sub-Saharan Africa compared to other parts of the world. And then there's the issue of the global data insufficiency as well. So at Revna Biosciences, we believe that given the institutional voids that we have, we need to look at things differently. We need to find alternate ways of addressing these issues in a way that is lean, in a way that's sustainable, and in a way that is relevant today and also for tomorrow. So on our side, we believe that the three problems that I mentioned are all interlinked. Africa's access to medicine and treatment failure, access to diagnostics or the lack of the diagnostic penetration in the region, and the global data insufficiency that we currently have in terms of diversity and inclusion of patients or people of African descent or Africans are all interlinked. And so looking at that when we decided to address the issue, what we did was to ask the question, what one solution could we find that simultaneously addresses all of this without breaking the bank, without losing the focus, and being relevant today with a view on tomorrow as well. So we decided to start from the basics. Institutional voids means we have certain technological, certain capacity issues that needed to be filled. So what we started doing was we focused on the infrastructure. We built a facility, 10,000 square foot facility for advanced molecular diagnostics, but with certain capabilities in mind, because we had the end goal in mind as well. One of the things we did was ensure that our labs are multidisciplinary, cutting across a range of specialties or subspecialties, from molecular to virology to bacteriology and all of those. We also ensure that from a trials and from a capabilities perspective, that these labs were equipped and agile enough to flex up and down depending on what is happening, especially on the backdrop of the last 2-3 years of the global pandemic. We wanted to ensure that our facility is a resource, because again, that's the point. You want to address issues in a way that provides relevance and provides some future proofing. We also are researchers at heart, and everything we're doing has that research capacity or research objective in mind as well. And therefore, in building our labs, we ensured that we had a fully equipped biobank, and we're playing at the international ISO standards 15189, which is the standard for medical laboratories, and then the 20387, which is the international standard as far as biobanking of samples were concerned. Now, after having the infrastructure in place, of course, we had to equip the place, and this is where the localization of technology comes in. So in the short space of time that we've been in existence, we've been fortunate enough to have some really exciting and involved partners who believed in what we're doing, who believed in us and invested in us directly and in diverse ways. Two of them are highlighted here. QIAGEN, a global molecular diagnostic provider, and then Diatec Pharmacogenetics as well, a leader in precision medicine assays and product development. We through these partnerships, and one thing I want to highlight as far as the localization of technology is concerned are two things. There's the equipment aspect of it, in terms of making sure that you have access to the newest and latest, but also the relevant tests in the region. But then there's also the capacity building aspect of it, and I'll touch briefly on each one. So in our partnership with Diatec Pharmacogenetics and QIAGEN, one of our stated goals from a revenue perspective is this: that all the tests that we provide locally will be tests that generally are not performed or conducted within the region, or if they are provided within the region, are tests that have unacceptable long wait times or turnaround times. So on the infectious disease side, we went to QIAGEN, had a discussion, and QIAGEN believed in what we're doing and invested in us by giving us access to the NeuMoDx technology. This is one of the newest technologies out there for random access testing of a lot of viruses or infectious disease targets. So you're talking about viruses such as you're doing HIV viral load testing, you're doing CMV, and a series of others. Currently in the region, these are tests that oftentimes take a few days to a week, in some instances a couple of weeks to be done. With the NeuMoDx, what we can do here and what we're doing here in Accra, Ghana currently, is that if you send your samples to us, anywhere around lunchtime, you can be sure that we're returning the sample results to you same day. That is the power of technology. That is the power of the collaboration we have with QIAGEN in one particular instance. On the oncology side as well, currently no molecular profiling is actually happening within the region. But by partnering with Diatech Pharmacogenetics, we're able to bring these tests locally and ensure that we can confirm, or we can tell our patients that, guess what? All these tests that are being done in Europe, we can do them here with the same assays and with the same equipment as well. So you're not losing out on quality, you're not losing out in cost, because we've had those discussions. And you, importantly, you're contributing to science, because we're paying attention to the trends, we're drawing insights from these, optimizing these tests to better suit ourselves, our population, but then also to spare discovery as well. Importantly, what we have committed to is that all of the tests that we run in our facility are either CE marked or US FDA approved, and also subjected to the Ghana regulatory process. What that does is that it helps us ensure the standards, but it also helps us contribute to the regulatory updates that are required, because through that, we're essentially challenging the regulatory authority to keep up with the trends out there, because we're bringing new technology, and they also have to be well-versed in understanding how these technologies work, so they can regulate us as providers and practitioners in the space. So the next thing I want to talk about now is what are some of these capabilities or areas and that we find ourselves in. At the moment, from a central lab perspective, given our multidisciplinary lab capabilities, we're able to serve as central lab services. This is an area where, in my previous life, we were scouting for entities on the continent that could provide these sorts of services, so that, given that a lot of the new therapies that are being developed are more targeted, meaning that to identify a patient that would most likely benefit from a treatment, you have to be able to understand at the molecular level what is driving that patient's disease. We're able to now provide some of these services to profile patient samples and diseases, and characterize patients in a way that can then go back to global biopharma and even diagnostic providers to say, we have this patient population here. How can we help them through access to medicine programs or participation in clinical trials to bring these products here in a way that benefits all stakeholders? Again, I mentioned the bioarchive capabilities that we have to be able to store these samples in a way that makes sense. One of the discussions that I had with Kafu, the next speaker earlier on today, is exactly in this space. How do we store these tissue samples to ensure that they are in the condition that will be useful in future research once the samples are collected from the patient? And again, we have invested heavily in this space to ensure that tissue samples that we collect, one, are collected in a way that is non-disruptive in the regular patient care continuum, but then importantly as well, that the starting material or the pre-analytical conditions of these samples maintain their utility when we come to do research activities with those as well. Again, collaboration with local partners is something that we've been actively working on since our establishment. And we're delighted to have folks like the Leiden Referral Center or Hospital in Ghana as part of our network, the Infectious Disease Center also as part of our network. But importantly, with all these partnerships, what it does is gives us access to a diverse patient population, large enough for us to be able to stratify our patients into the various subtypes that are required, to then say, these are the studies that these patients would most likely be able to participate in a meaningful way. And then through these participations, we can then start looking at how do we optimize existing therapy? How do we then start thinking about drawing insights from these to spare the next generation of medications, treatments, therapies, and all of those. And through these local partnerships, we're then able to expand the reach of what we all communally do or can do in this, because we are then pooling our resources together, and as a result can then tackle multiple of our diseases or disease areas together simultaneously. Additionally, and on this slide, I just want to touch very briefly on the spectrum of partnerships that we have. As you can see, we have academic and healthcare partnerships that I have mentioned. We have technology and digital solutions partnerships as well as partnerships that help us extend our reach on the continent as well, because ultimately the goal is to pool these resources or the resources that we have that may seem disparate to ensure that the patient impact and patient coverage is optimized. So a few current points in terms of areas of specialization. Because of the approach we've used, we're able to play in the advanced molecular diagnostic space, providing unparalleled turnaround times, providing molecular profiling locally, as well as providing biobanking capabilities that hitherto were not available in the region. Central lab services I have mentioned, and then the translational and clinical research capabilities that we need given the current focus of, or the current trend of more targeted therapy requiring certain analytical procedures being done locally in particular to ensure that patients are classified properly and matched properly with the existing studies and therapies as well. So currently, based on the model and the activities we've seen, I'm just going to run through this slide quickly and then show one last slide on the impact that we have. The immediate impact is evident in the sense that we're at this point addressing those unmet diagnostic needs locally, profiling patients' tumors, and providing insights useful by oncologists and patients in determining which treatment options to go for and why. The model also allows for that sustainability thing, which is an issue in the biomedical space, especially on the continent. Because if the model isn't sustainable, it's going to crumble, and then it becomes a disincentive and causes other challenges in terms of fundraising and attraction of other global partners to come and play in this space or support local enterprise in this regard. And therefore, for us, sustainability has been a key point. Real world evidence in the sense that given the way we've digitized our solutions and the ability to recall patients, we're able to draw insights or glean insights that can then be incorporated into the existing medical and molecular diagnostic insights that these patients have. But then we begin to see how they're faring in the real world to further enhance what we do. The biomedical and clinical agility part, I think, has been demonstrated. So I want to end by showing one of the things that we're doing, or one of the things that areas of activity that we've engaged in. So once we got the facility out of the gate, the next thing was to bring these oncology tests that up until now were not being done locally. Now the challenge with that is that you're in a situation where these tests are not locally done. We have committed that all the tests we bring have to be CE marked or US FDA approved to ensure their quality. But importantly as well, that we wanted to subject these tests to the local regulatory scrutiny as well. All of these, one, to build confidence in what we do, but then also to help with the advancement of the regulatory system that we operate in. Now, to do that, we had to be innovative. And thankfully, the Ghana FDA was very flexible and accommodating in hearing and working with us to ensure that we were able, or we were able to come to a pathway to bring in these products to the market quickly in a scientifically rigorous way, while maintaining the regulatory standards that need to be applied. So in between December and now we've had our first clinical study approved, the protocol approved. We've gotten Ghana Health Services ethical clearance and some other institutions clearance. We've gotten the study underway. Last week we received a total of just under 30 patient samples to profile right here in our facility in Accra, Ghana. And as you can see, this study is designed to answer scientific questions while addressing patients' needs concurrently. It's also, especially if you look at the third point, it's also positioned to help us catalog the patient samples in a way that can help, or that would help with future research as well. It's a big samples study, 600 initially, potentially increasing to about 1,000, and we're looking at multiple cohorts as we go along. So these are some of the approaches we have taken. These are some of the steps we've taken in ensuring that we're addressing the biomedical institutional voids locally. We're also addressing patients' need right now, but then keeping an eye on the future. We're also conducting clinical trials right here locally in a way that ensures our participation in ongoing drug development, ongoing diagnostic development as well. So I'm going to pause there, and then we'll leave the rest for the Q&A session. Thank you so much.
Thank you. Thank you so much, Dr. Akwalu. I think that you have aptly demonstrated to us that even though you are working in an African context, you can adopt values in service delivery that commit to quality, but also in terms of bringing advanced molecular capabilities locally, and still committing yourself to high quality standards as well, both local and international. Now, your last slide partially answers one of the questions that I was going to pose to you, and I would like you to start thinking about it, is that obviously your problem statement in your presentation speaks to this conundrum of having to import 95% of products, and yet we're producing only 3%. Now, importing technologies could be part of the solution, and I do not doubt that at all. But I think that we also need to think about, if we are going to import, exactly what is it that we are doing to ensure that we are optimizing and adapting those technologies to the local context, including matching that with the local genetic diversities that we have. So I can see how that process of having to pass through the local regulatory processes could demand of you to prove that the technologies you're bringing are relevant. But I would like you to think about how you could, I suppose, expand on that question of how do we go about doing that? Because I was thinking that part of the solution would be to support local manufacturing, to ensure that we are producing de novo products and assays that are formed out of our local genetic diversities, right? But in what you are saying is that actually you could still look at the gaps in terms of advanced technologies and bring them locally. Now, that process of trying to reconcile those two issues would be important for you to think about. So if you can address that, that will be wonderful. Just keep thinking about it. But without further ado, let me invite Professor Patrick Kafui Akakpo who is an associate professor of anatomical pathology at Cape Coast University. He's very passionate, he's a medical doctor, very passionate about molecular pathology. And he's going to talk to us about the role of pathology in molecular diagnostics and how research can facilitate precision oncology. Over to you, Prof.
Thank you very much for having me today. It looks like my work has been made easier for me because the two previous speakers have gone into detail about what this entire meeting is about. I do not have any disclosures, and my topic today captures a number of things. One, it shows that there's a central role of pathologists in the diagnostic process when it comes to precision oncology. So pathologists need to be abreast with new things to be able to initiate the processes involved in precision oncology or treatment. Okay, and in doing that, pathologists also become allies to tissue banking and research. And I am interested in discussing the role of the pathologist in 5 broad areas: molecular diagnostics, as it feeds into precision oncology; genetic testing, again, as it feeds into actionable mutations that are picked up, whether it is in relation to treatment or it is in relation to preventing cancers from occurring. Then I would also want to discuss the role of pathologists in relation to research, modern research, and how they can collaborate with people, other researchers, to get this research done properly. Then I would want us to look at the subject of real-world data, ongoing data, which is collected the right way to ensure that it feeds into RCTs when we start having randomized control trials, or better still, what we call pragmatic randomized control trials, which kind of simulates the real world environment. And that is actually what we want to have real control trials look like. And then, of course, I'll go back into the issue of tissue banking. What is different now? What we need to do now to improve it to support the precision oncology and research that we need to be able to be part of these clinical trials. So, molecular diagnostics. The baseline when it comes to precision oncology and clinical trials is that you need molecular data or molecular diagnosis to be part of any of these trials. So it means that the diagnosis should go beyond the usual things that a pathologist does, which is H&E special states and basic immunohistochemistry. You would require a topper to be able to be part of any of the clinical trials. What it means is that your diagnosis should be that specific molecularly to be able to be part of any clinical trial. So what is it that we are interested in if we are not interested in basic H&E, special stains, and basic IHC? We are interested in things like PCR, NGS, whole exome sequencing, and to the end, complete genomic profiling. So you get to understand the molecular basis of the disease. Now, IHC can serve as a window to what I have said. You can use immunohistochemistry as a window to determine certain mutations, and there are specific markers that are approved by FDA for such purposes. So even before you start looking at, let's say, HER2 for Herceptin, you can do immunohistochemistry, and if that fails, you then escalate to ISH, and then you can move on. The same with mismatch repair. You can do immunohistochemistry as a window to mismatch repair, and if that doesn't give you the information that you want, or it gives you a lead, then you can proceed with further testing. And there are many more I can list, okay? So molecular methods are employed in diagnosis to enable molecular therapy or other action based on a patient's molecular profile. Now, when I use the word action, what I mean is you find a mutation and it can be actionable. So what action can you take? That is treatment, a drug-based treatment being part of a clinical trial, or sometimes not just that, but preventing other cancers because you found something that makes the person susceptible to multiple cancers of different sites or recurrences. And that is also an action. Then again, there are people who are related to the person, and you may have to also prevent them from getting the disease that the index patient has already. Okay? So it allows for diseases to be studied properly so that you know their molecular signatures. And that can help you treat them now and follow them up, and also prevent others from coming. And it ensures that the evolution of tumors can be predicted and identified. And it allows for novel traits or features to be determined and actioned. And again, it provides the baseline molecular treatment, molecular information, which is what I said earlier, for recruitment into any of these trials or actionable processes or procedures that you want to engage. Now, genetic testing is something that appears not to come up when we start talking about diagnosing a patient, but it is important if you want to do molecular treatment or precision oncology, because we are looking at things at the molecular level, and there are mutations that may not just be present in an index patient, but also in their family. So it's a germline mutation, it runs in families. And that becomes a critical thing that pathologists must be able to pick up. So what it means is that when a pathologist is looking at a slide and making a diagnosis of, let's say, cancer in a young person, the pathologist must be aware that cancer at a certain age requires further investigation to be able to rule out genetic basis. Okay, so you get a cancer through a number of things. You can say, okay, if I get this type of cancer in this particular age, I would have to test the patient for certain genetic malformations to be certain that this thing doesn't run in their family. And if it does run in the family, there are a number of steps that you can take, which would include the patient and the patient's relations. So that also becomes a cardinal role of pathologists. Pathologists at this point will serve as a kind of central or gatekeeper that would reflect or prompt other people in the molecular diagnostic process or molecular management process to keep an eye on the patient or the patient's relations. So genetic testing allows for current and future treatment. And it also ensures that you can plan treatment for patients or their susceptible, their relations. And this is in line with a patient who has an index cancer or a cancer that you can predict would come or would a patient will be susceptible to. So as an example, we can use Lynch syndrome. If you do have an endometrial carcinoma that is of a certain grade, that shows certain features on testing, you can predict that the patient may have Lynch syndrome, and it will require that you do genetic counseling and testing to be able to prevent the patient from getting other cancers like colon cancer. Okay, so by looking at endometrial cancer, you are now looking at colon cancer, and in the same way, other people in the family who have that syndrome, if tested and confirmed, would also be predisposed to the same cancers. So that's the idea of genetic testing, and pathologists must become aware of these things. And I'm only using Lynch syndrome. There are many other cancers that are also going to present as cancer of multiple sites, cancer at an early age, cancer of a certain grade. And pathologists must be aware of this. That's their modern role. It becomes a very important modern role for pathologists. And they are amongst the few people who can actually start the process of this genetic, of this molecular testing and eventually clinical trials. And then you go to precision medicine or precision oncology. Now, research. Pathologists' role in research has not been in doubt, but we are not talking about basic research that is just collecting epidemiologic data and type of tumor and all that. In this scenario, we would want the research to be at a certain level that requires collaboration, okay? And with that, it has become even more imperative that pathologists collaborate, especially because we are part of what they call MDTs, or multidisciplinary team. And once you are part of it, you would have to collaborate with many people. And it's not just related to just other clinical colleagues, but scientists, data scientists, and research scientists, and geneticists, and all that. Okay, so pathologists become central in carrying out even the randomized control trials that we are talking about, or pragmatic randomized control trials, or whatever form of research that we want to take. And the modern pathologist must have the expertise to be able to play that central role, not just in picking up people who qualify for such trials, but also in ensuring that those who are picked have the right molecular data to be part of such research processes or such clinical trials. Short of that, the recruitment process will become a very cumbersome process that may result in a lot of waste. And RCTs are already very, very expensive. And so if you don't do it well, you're not going to benefit from that. And ultimately, you will not benefit from the applications. So in both clinical and other research, pathologists must be able to provide skills that enable the process and ensure that now we can be part of this precision medicine. Now, real world data is the other thing I wanted to talk about. And what is real world data? Now, this is data from sources other than traditional scientific research, but I use that guardedly because other types of research are also scientific, but generally we prefer to refer to those that occur in a controlled environment as scientific research. And anything out of that, a randomized control trial or a specific research, you would consider as real world data. Now, in relation to medicine, there are many things that qualify, but pathology becomes one of the most important places where you can collect real world data. And there are advantages that we must clearly benefit from, or things that we must take advantage of as pathologists. And the modern pathologist must be interested in ensuring that certain data is captured, some kinds of data are captured, and that data is robust enough to be used to inform clinical research or any form of scientific research for that matter. Now, what do I mean by that? If you look at molecular data, that's molecular profiling, it is one of the sources of real-world data. And that eventually becomes a part of real-world evidence that can inform even the clinical trials that we are doing. Again, if you look at the pathology report itself, there are many things in the pathology report that can inform real-world evidence because it is real-world data. And these things are in line with, they relate to the diagnosis, the grade of the tumor, the stage of the tumor, the immunohistochemical profile. These are very important things that can be captured in a robust way. And that data becomes an important source of data that informs the new things that we want to do, the molecular-driven treatments or management procedures that we want to carry out. Now, there have been traditional issues with real-world data in relation to pathology. The quality of the data has been very porous. It is almost dependent on the pathologist and how well he's able to speak English, because it's a narrative form. He just starts writing and then he goes on and on, trying to justify why he made that diagnosis. The question is if that justification is important or necessary. What is necessary for the treatment of the patient? And the answer is no. What is required to treat a patient is what we call core data items. How far has the tumor gone? How aggressive is the tumor? What is this treatment tumor going to respond to? Basic things. Has it gone into the lymph nodes? Has it gone into the lymphovascular space? Is it likely to show metastasis if left alone? So these are things that are important, and data must be specific for those. It must be captured in a way that is robust to allow those things to be retrieved when necessary so that it can feed into clinical research. So all the problems aside, if you look at the real world data, the thing that what is the advantages, and of course it represents the real world setting. You cannot use a controlled environment and compare it to the real world. Your controlled environment may not work in the real world. So getting the real world data also becomes very important as it relates to real world evidence. And pathologists must be able to provide that, irrespective of the fact that a randomized control trial is going on. The data and evidence generated by a pathologist must be robust enough. And that's the role of a modern pathologist. Of course, it's also cheaper. You can put a cohort of people on a certain medication. Herceptin, as an example, is a medication that is taken by millions of people around the world now. What it means is that if the data is collected properly, you could actually determine newer side effects and then doses and all that were not possible within the randomized control trial period because it was a controlled environment and the cohort was not that large if you compare it to the size of the population that will be on it in the real world setting. Then, of course, you can easily integrate this, what I'm talking about, into randomized control trials. In fact, it prepares the grounds for you to start the randomized control trials. Without that, you'd have a lot of teething problems and it will become more expensive for you because you can't go through the process of the RCTs well. The recruitment will become a problem. So it's important that we look at we look at real world data and real world evidence. That is big data, and that informs the control trials, which are in smaller cohorts of people and give us the environments that we want to create to be able to know what a drug will do and how people will benefit from it. Now, real world evidence is this clinical evidence that is about potential benefits or risk of medical products derived from real world data. So real world data feed into real world evidence. And a quality report is what I have been talking about since I started. A quality report means that that report is accurate, it's complete, it's timely, and it is usable. And all the data elements that are considered core data elements are captured. Once that is done, it sets the ball rolling for the more complicated data that is going to come out of molecular studies and molecular testing. Now, this is a good example. Real-world evidence to support therapeutic development in hematologic malignancies. It's not very complicated. So the data will be generated, evidence will come out of it, and you would know that you are developing a drug, and these are the things that are associated with that drug. So it's very good data if captured well, and it informs what we want to do in relation to molecular therapies and then precision oncology and clinical trials. Now, there are clear things that must be done. There must always be a paired data element and response. It allows the data to be retrieved in a way that is simple, something that can be exported immediately to Excel, SPSS, or any other format that you want it. But you must make a conscious effort to capture the data that way. If you have to describe the data in a way that you have to fish out the difficult or the important things, it's not going to cut it. And modern pathologists must be aware of how to enter their data in relation to the diagnosis that they are making. It will get even more complicated when we start doing molecular therapies, when we start doing molecular testing, because the data that is generated is a lot of data. And without a robust way of capturing it, storing it, and then assessing it, it immediately becomes unusable and we would not benefit much from it. And pathologists must be able to play that role in the modern sense. Otherwise, they are going to be people who would put impediments in the way of the RCTs when it starts. Now, this is an example of a tissue bank, which is one of the things that I want to talk about. You look at it, this is formalin fixed paraffin embedded tissue, and it's in blocks. Okay? And traditional pathology labs have always had this form of storage. There are clear disadvantages of having this. This is real world material, okay? And you have to start from the get-go thinking about molecular pathology. If you don't do it that way, these blocks will be useless when it comes to RNA or DNA work. You can't get much from them. What it means is that you must store the tissue, you must preserve the tissue in properly buffered formalin, 10% buffered formalin, right volume, the duration must be right. That way, the tissue is prepared for molecular testing. If not done well, you would not be able to get any results from the molecular testing, and therefore, you cannot even benefit from the RCT that you want to. So pathologists become very important when it comes to data from tissue banking. And it is not just related to formalin fixed paraffin embedded tissue. You would have to look at things like fresh tissue. You may have to look at frozen tissue if it is fresh, and other forms of freezing that ensures that molecular material is robust enough for molecular testing. So in that regard, the modern tissue bank would look at fresh tissue, mostly tumor, sometimes normal tissue, blood, because you do tests that are related to liquid biopsies and you give you a spatial understanding of the tumor and not just a localized understanding of the way the tumor is, if it is just in the breast. Blood and other tissue, other fluids can allow you to look at the thing in relation to the entire body. And again, it can also help a lot with mutations that predict how a tumor is going to behave. Then, of course, you've got the traditional formalin fixed paraffin embedded tissue, which we have to now learn to prepare properly so that it is robust enough. And again, when we prepare it, we must immediately make it available through collaboration for research, because if the tissue is kept, formalin fixed paraffin embedded tissue is kept for so long, it is going to become irrelevant, because after a certain time, it is no longer stable anymore for molecular research. So whilst pathology labs begin to believe that by keeping it for 10 years, 20 years, it is still good enough, if you haven't prepared it properly, even after 5 years, it's not going to be of any use. So you must collaborate to ensure that the tissue that you're collecting and banking is not tissue that is useless just after a short period. And you don't also keep it for too long, because if you keep it for that, it eventually does not make any sense anymore. It will not be useful. Then, of course, we need a supporting clinical data, and you need the pathology data. And I've said that real-world data in relation to pathology becomes very important. And modern pathologists must be able to capture data in the modern way that allows real-world data to be used to feed real-world evidence. Of course, you need mandatory equipment, fridges, liquid nitrogen, and many other sophisticated things, which Derek has already mentioned. And pathologists must become adept at using these methods to store tissue for molecular testing that will inform clinical trials in precision oncology. The collaborative effort is important. Without it, it becomes something that pathologists alone cannot do. Now, there are other things that I believe may come up during the question time, and I'll be open to answer those during that time. Thank you very much.
Thank you so much. Thank you so much, Patrick. Much appreciated. You can take down the slides and maybe just put on your cameras once again, colleagues, as panelists. I want to thank you all for the talks that you have presented. And Patrick, much appreciate your talk, delving deeper into the application of precision medicine, but also, again, the common theme of multidisciplinary team collaborative approaches that keep emerging, especially in terms of instant use of samples, but also instant use of data. And in as much as data may be complex in the form of big data, but your emphasis on issues of storage, I think both in terms of tissue storage, but also data storage, it's important. You mentioned analytics, which means we must also be thinking about capabilities for analyzing real world data, complex as it may be. So all of these things speak to a value chain of different skill sets, different infrastructure that needs to be in place for us to be able to holistically do this work around diversity, especially if we are going to make sure that, as you've already stated, that we do things promptly, immediately, and we are able to provide that immediate service, as we discussed. I want to come back to some of the issues that I've raised. Maybe we're going to start with you, Kemi. If you can comment on the first issue that I raised around mistrust, but maybe let me add a couple of other issues. There is a point you raised around the patient-centric approach that requires an understanding and an incorporation of cultural nuances in the way that we approach clinical trials, which then speaks to cultural diversity. Cultural diversity as it may be linked to genetic diversity, but also in itself, in and of itself. You mentioned this issue of economic status as well, that it may affect issues of transportation, access, and so forth. I would like you to kind of help us, those people who are coming from a molecular biology perspective, how can we have an appreciation of those issues that we consider social sciences, right? Around culture, around economic factors and so forth, as we navigate this issue of diversity in clinical trials. There was also a point, so those are my two issues for you. There was also a point that was raised in the Q&A, you may have seen it, and it speaks to current collaborative efforts that may be happening on the one end, and then on the other end around capacity building, often geared towards investigators and coordinators, and maybe in some cases communities. But in general, it doesn't really speak to the foot soldiers, and we may be missing opportunities there. That's one of the questions that was raised in the chat. So let me leave you with those and then hear your thoughts.
All right, thank you. Very good question, actually, because most times I'm a scientist by my academic background as well. So most times when we're designing clinical trials, we're looking at scientific factors, right? You want to enroll patients with a certain blood pressure level, right? You want to account for or at least avoid confounding factors. So maybe they shouldn't be on one medication if they are going to be part of this clinical trials. We'll look at it mostly from the scientific end of things. And most times also we look at it from the regulatory end of things, right? Do you have informed consent before you enroll the patients? But when we start looking at taking research designs in other parts of the world and bringing them into Africa, we need to start considering cultural requirements that can influence how well those studies are conducted. So for instance, right, if you have a trial that has as part of it the need for women to be on birth control pills, well, depending on their religious background in Africa or their cultural background, it might influence that particular criterion, might influence the ability of the woman to join the study. If he has a husband that culturally does not believe in that, or by religion does not believe in that, that could be a very ideal candidate for a study that we may have ruled out, right? So one of the things that we should start looking at is, what is the context in which we want to run clinical trials, and are there possible modifications? There are instances where you can modify, right? If you need patients to be 65 years and above, enrolling a 50-year-old patient may not be ideal. But we should start looking at trial design so that we are accommodating some of these nuances without jeopardizing the scientific integrity of the trial. So this is an example that I'm giving in terms of how we, take into consideration nuances. But I want to talk about socioeconomic factors, because that's not even limited to Africa. Even in the US, right? If you have patients who do not access the healthcare system because maybe they did not have insurances or they did not have money to pay out of pocket, which in the case of Africa, many people are paying out of the pockets, right? You could have very, very eligible patients out there that you cannot recruit because they're not even showing up at the doctor's office enough for the physician to be able to say, would you be interested in this trial? And so now we have to start looking at, how do we take trials to them? How do we make that person, that market woman, right, aware that there's this clinical trial for blood pressure that she may actually be an ideal candidate for? And so how are we encouraging ourselves to pull in people who do not normally access the healthcare system because of their socioeconomic status? And in fact, that takes a lot of collaboration as well across many stakeholders, which is why I really continue to hammer on having collaborations as a way to address many of the issues that we suffer when it comes to doing clinical trials in Africa.
And can you comment on the collaborative efforts?
So, the great thing is, look, this conversation we're having is part of what we call collaborations, right? I'm from a different background, you all are. And so this is one of the things we need to start talking. We're not just, I guess what I want to emphasize is, we're not just talking about it, people are already doing it. I know of the European and development countries clinical trials partnership. That's a group already that is linking developing countries investigators and with European countries investigators to design trials appropriately, to support trials appropriately, to fund trials appropriately. We have the BioVenture BVGH. I don't want to call out the name incorrectly, that is also a non-profit organization, actually, registered in the US, but supporting a lot of oncology clinical trials activity, pulling stakeholders together to have conversations around how we find patients, how we enroll them, and how we keep them, and how do we keep sites, eligible or qualified sites so that they're able to be part of clinical trials. So one thing that we should all know is conversations are happening. Partnerships are happening. What I would like us to start to do is to start talking a little bit more about what is happening so that people can actually see that we are taking actions. And then the next step is to start measuring results. So I'm hoping that the next time we have this webinar, now that we've talked about the frameworks and solutions that are being implemented or that need to be implemented, we can start now checking to see which of them is successful. But I saw somebody else on the question too, that was asking about his partnerships. I'm willing to provide you some listing of different organizations that I know have been set up really to help drive research and development and clinical trials activities in Africa. And I'll be willing to share the list with people. So they are there. We just need to continue to engage.
Kemi, can you comment quickly on the parachute research and its harm, and whether we will be able to navigate this going forward, given the context of mistrust that we are in?
So, like they said, necessity is the mother of invention, right? So the reason I say that is Africa is not the only part of the world that has suffered from some of the poor decisions, that were made in the past by sponsors, right? We know what happened even in the US, right, with the Tuskegee Airmen. And so, but the fact that something bad happened in the past should not get in the way of our ability to start doing good in the future. So yes, there were many instances in the past where we had parachute research happening. People come in here, do the trials, gather their data and run away. And even the people that contributed to it, whether patients or investigators, did not benefit from any of those trials. But I can tell you, based on engagement across multiple stakeholders, we know that that narrative is changing. I don't want to say it has fully changed because we still need to measure, but we know that there are rules now that are in place. Regulatory agencies now are, are putting in place guidelines around good clinical practices. We are more informed about what we should expect from our external stakeholders. We now have some training within the continent. We still need more so that people now ask the right questions. What is going to happen to that sample, right? Who has right over that sample? Where are you going to store it? And so the narrative is changing. And I think it is important for us in this field, where we're looking at how do we bring new therapeutics and diagnostics to the market to enable better healthcare in Africa, to also help to start driving that change. We need to start informing people about the positive benefit of being part of research and acknowledge that mistakes were made. That yes, that those have created mistrust, but also acknowledge that it is time now to move on and to start making strides to achieve objectives and goals that are related to research and development within the region. So yes, I think there's improvement in that aspect.
Thank you. Derek, your point about localizing technologies, Kemi just mentioned that we do want to bring those advanced technologies, but how do we make sure that they are suitable locally and that we don't cause any harm and that people can trust them? What do we put in place? Your comments on that?
All right, thank you. And so with regard to localizing technology and ensuring that they benefit the communities or populations within which they're being localized and deployed, one of the things that we're looking at, especially in the context of diagnostics or advanced molecular diagnostics, is exactly that. That is one of the reasons why we have committed to subject all these tests to the regulatory scrutiny of for instance, the Ghana FDA. And one of the things we're trying to do here is this, to understand that in order for us to do these precision medicine related studies, activities, manufacturing, and all of it, there's one key point that we all have to be cognizant of right now. Is the lack of a baseline data. Now, how do you generate that baseline data to then build upon that? That is where we're saying, let's use technology to address the gulf between what is being delivered now and what is acceptable or what is available globally as the best available option. Now, in doing that, two things we want to keep an eye out on. First is that we have patients today, and we're going to have patients tomorrow. But we want to care about both sets of patients equally, right? So in the first instance, how do we bring this technology here to ensure that patients today benefit from those technology? And how do we use those experiences and the availability of the localized technology to ensure that we're planning the future by ensuring that we draw insights from the existing situation to begin to put things in place, either to intercept diseases or to be effective or better able to manage those conditions down the line. So that's how we're using the technology. But then importantly, one of the things we're doing as well is to say we're evaluating these on a case-wise basis and say, for instance, we brought in a 23-panel respiratory pathogen test to West Africa. Are these 23 pathogens or bacterial virus combinations even relevant in West Africa? We can only know that after applying them, and again, talking about what Kafi was saying, incorporating those real world insights into that to then say, hey, guess what? Of these 23 pathogens on this panel, maybe we only care about these 18 because these are the ones that are endemic in this region, and then we optimize. So it's that iterative process of introducing the product to the community, paying attention to ensuring that we're meeting today's patient needs, but then asking the question to say, how do we better improve these existing technologies, tests, and medicines for the next patient that is coming down the pike as well. So that is the duality of what we have to do to ensure that these technologies are useful today and then serve good purposes for tomorrow's patients as well.
Okay, thank you. So, I mean, I hear you, and I think that in a way, it will be good in future discussions to start talking about the results from those evaluations so that we can demonstrate how we are modifying them or adapting them, as well as increased levels of acceptability as people begin to respond to them. But Patrick, there's one question for you, and it's got to do with the fact that in as much as you've made a good argument around real world data, it may not be of interest to biopharma, big pharma, for example, but it may be of interest to those of us who are in Africa. Now, do you believe that there is a sort of interest in this from a big pharma perspective, or is this something that needs to be built bottom up in order to ensure that the success of diversity in clinical trials is informed by the use of real world data from within Africa?
Okay, so thank you for that question. I must say there is no doubt about it that Big Pharma is interested in real world data and real world evidence. Many of them have made it a point to find that data and make it more robust. However, I would not consider them as important in the argument for real world data. I would consider ourselves as people, okay? Now, what we need to do is to have data that tells us what has been happening to us in terms of diseases. And even that is a problem.
Yeah.
The reason it is a big problem is if you take a pathology report or any other report related to cancer of a patient, it is very difficult to get what we call the core data items. So I'll use basic examples. You need a type of tumor, the grade of the tumor. You need to know if immunohistochemically, like a breast cancer, is hormone receptor positive or is HER2 positive, because this is data that is important for the treatment of patients. Now, today, every patient would have their report, so it makes it look easy. Next year, at the end of the year, we want to know how many patients are HER2 positive, because we want to make an argument to Roche that we've got the numbers in Ghana for treatments to go on health insurance. And then don't have the data because we never kept it well. So we can't make an argument to pharma to even give us the medications. And then you notice that they are not interested because you don't have the numbers, you don't have the evidence. So it is in our interest to start the process bottom up. Of course, it will become important to them because they would get to realize that the numbers add up. And so then it becomes important to them. But elsewhere, they have already become very important. In real-world data and real-world evidence has become a focus of big pharma movement, especially when it comes to pragmatic randomized clinical trials, because that information helps them to better plan the randomized clinical trials to simulate the real-world environment. Otherwise, they do it and then they have to terminate the trial midpoint because they didn't recruit well. They didn't have enough information about the real world before doing the recruitment. And we see that all the time. So it is actually important that we start ourselves. We will get help, and the help will come very, very fast if they realize even the little numbers add up. That's my opinion. Yeah.
Thank you very much, colleagues. Ms. Williams had to drop off. We are out of time, 5 minutes overdue, but I want to thank you all for the presentations and also for the questions that participants have raised as well. Thank you for those messages. We are getting the message, Patrick, that we must not just let Big Pharma be the one to set the agenda. We need to start thinking about how do we drive that agenda bottom up as well? And much of this collaborative efforts is something that we need to think about. And I think the message is clear to the organizers as well, that I think going forward, we really need to think about a different constitution or composition of the teams that are going to drive this agenda. They need to be diverse. They need to come from different sectors, and they need to bring different sets of skills. It mustn't just be the conversations that emerge only from a molecular biology perspective or pathology perspective, but it must also start to talk about the regulatory aspects. We must talk about how people who are dealing with routine data and routine data sets can also be part of the conversation so that they can understand the importance of that information in how it starts to drive the agenda, to drive the demand side of things. But of course, also having the capability to evaluate these technologies that we bring locally or that we develop locally so that we can generate the evidence to demonstrate how they affect us, how they affect people, how they are taken up, and also continue to make sure that they can be trusted and used. So let me stop there, colleagues, and thank you again for participating and wish you well for the rest of the day as well. Thank you very much. Thank you to the organizers.
Thank you too for having me.
BioAfrica and Amgen. Thank you. Have a good rest of the day.